Okay, so welcome to this next video in which we are discussing the anthrax toxin. Okay, so we are currently in the process of discussing how the anthrax toxin, protective antigen 83, is going to bind to receptors on the surface of cells, basically. So we've discussed the receptor, well, we've discussed the two receptors, but we're focusing on a single one of these, which are the tumor endothelial marker 8 protein and also the capillary morphogenesis gene 2. Now, as I say, we're going to focus on tumor endothelial marker 8. And tumor endothelial marker 8, which is shown here, has a binding site for the protective antigen 83. Then it has a single membrane spanning alpha helix, and then it has a cytoplasmic domain, and on the cytoplasmic domain 2 cysteine residues have been palmitoylated, and these palmitoyl groups are anchored in the inner leaflet of the phospholipid bilayer and stabilize the tumor endothelial marker 8 protein at the plasma membrane. Okay, right. Now, there is a co-receptor, basically, for the binding of protective antigen 83. So, this is a protein which binds to the tumor endothelial marker 8 protein and is necessary in order for the tumor endothelial marker 8 protein to bind to the protective antigen 83, although it itself does not form the binding site. However, if you don't have this co-receptor bound, the receptor TEM8 will not bind to the protective antigen 83, so it will only work if you've got this co-receptor here. So that's the definition of a co-receptor, a molecule that is necessary to have bound to the receptor in order for the receptor to actually bind to its ligand, basically. Now, what is the name of this co-receptor? Well, again, it's got a single membrane spanning alpha helix here, and it's called LRP6, okay? And, whoops, and they put a G there, LRP6. In fact, I did put a G there. LRP6, okay? And LRP6 stands for LDL, or low-density lipoprotein, receptor-related protein 6. Now, where am I going to write this? I'm going to have to put it down here. So, the L is for LDL, which from, oops, let me bring this up. Okay, and remember, LDL stands for low-density lipoprotein. Okay, so L is for low, D is for density, and then the other L is for lipoprotein. Right, so this first L of LRP6 is for LDL. Then R is for receptor-related, okay? So this is, both of these words sort of have a dash between them, okay? So they are abbreviated to a single letter in the LRP6. So the R is for receptor-related, and then finally the P is for protein, and then the 6 is for 6. So this is the LDL, or low-density lipoprotein, receptor-related protein 6. Okay, so let me give this a little bit of colouring in. So we'll have LRP6 in turquoise here, and we'll have TEM8, or the tumor endothelial marker 8, in red here. Okay, right, so we're now ready, we're set up, we have our receptor for the protective antigen 83 all set up. All that needs to happen now is that this receptor needs to get exposed to the protective antigen 83. So, basically, if you're exposed to the protective antigen 83, what's going to happen is it's going to come and bind to the binding site on this tumor endothelial marker 8 protein, and we'll go from there. Okay, so let's draw this out again. So here is our tumor endothelial marker 8 protein. Here is our LRP6 co-receptor here. And of course we need our palmitoyl groups coming off the cytoplasmic domain of the TEM8. Okay, so then what's going to happen is the protective antigen 83 is going to come along and bind to the ligand binding site on the tumor endothelial marker 8 protein. Okay, so here is the protective antigen 83. So it's come along now, and remember the protective antigen 83 is about to be cleaved into two parts, the protective antigen 63 and the protective antigen 20. Okay, and we're now going to see the enzyme which is going to catalyze that, and this enzyme is going to be within the plasma membrane. Okay, so this blob here can represent the enzyme which is now going to cut the bound protective antigen 83 into these two parts. Okay, and this enzyme is called fucin. Okay, or fusin. Right, now fusin, what fusin is going to do is it's going to break protective antigen 83 into protective antigen 63, this big fragment here which will remain bound to the receptor, okay, and it's also going to break it into protective antigen 20. Now, as I've drawn, protective antigen 20 isn't in contact with the receptor, so basically it's going to go off. Okay, so what's going to happen is that you're going to cleave the protective antigen 83 into the protective antigen 63 and the protective antigen 20. Okay, and what's then going to happen is the protective antigen 20 is going to go off into the world, basically. It's going to go off and protective antigen 63 will remain bound. Okay, so to demonstrate this, I think I should colour in the two separate fragments in different colours. So, here is protective antigen 20 in blue. So I'll label that up. So this is PA20 for short. And the one that's remained bound to the tumour endothelial marker 8 protein, that is protective antigen 63. Okay, so I'll have that in green. And now I should also colour code the same bits in the same colours over here. So, the portion that will make protective antigen 63 is in green, and the portion which will make protective antigen 20 is in blue. Okay, so this is protective antigen 63. Right, now, once you've got this complex of protective antigen 63 with the tumour endothelial marker 8, whoops, nearly went TUM rather than TEM, TEM8, okay, and then also the LDL receptor related protein 6, LRP6 here, what's going to happen is you're going to get multiple of these forming in the cell membrane, and they're going to start oligomerizing, they're going to start sticking together, and specifically they're going to stick together in groups of 7. Okay, and when you oligomerize 7 things together, that's called heptamerization. So what's going to start is heptamerization. So you're going to start forming heptamers. Okay, so how's the best way to show this? I think I'll try and show it. Okay. In fact, no, I'm going to show it just as a great big complex. So, and then I'll talk you through it, basically. So, what's going to start happening is you're going to take 7 of these things. You're going to take 7 of these complexes where you have PA63 with tumour endothelial marker 8 and LDL receptor related protein 6, and you're going to join 7 of them together. Okay, so there's 1, there's 2, there's 3, there's 4, there's 5, there's 6 and 7. Okay, so you're going to take 7 of the things together, and you're going to glue them all together to make up this great big complex, basically. So each one of these pieces is a whole one of these, basically. So let me try and make this even more transparent. So one of these purple pieces here that I'm now outlining in purple, this represents all of this. Okay, not the PA20, that's gone off. Okay, but the PA63 with the tumour endothelial marker 8 and the LRP6, all of that is together as 1/7 of this heptamer here now. Okay, right. Now, what's special now is that this complex is now capable of binding to the other 2 components of the anthrax toxin. So remember, the protective antigen was only one of the proteins that was being secreted by this cell, as well, by this bacterial cell, and was a component of anthrax toxin, but there were two other proteins that were also being secreted, the edema factor, EF, and the lethal factor, LF. And now these factors can come and bind to the extracellular side of this heptamer here, and 3 of them can come and bind. So let's show this. So let's have this as 1, here is 2, and here is 3. So there are 3 binding sites. Now, it's not specified how many of each you have, basically. So there are 3 binding sites, but you could have 3 LFs come and bind here, or you could have 3 EFs coming and binding here, or you could have a mixture, maybe 2 edema factors and 1 lethal factor, but it's not specified strictly how many lethal factors and how many edema factors you're going to bring in. Overall, you will bring in 3, but whether you want to choose to bring in 3 edema factors, i.e. whether 3 edema factors bind here first, or whether 3 lethal factors bind here first, or whether 1 lethal factor and 2 edema factors bind here, basically it doesn't matter. Okay, so these are just binding sites which can bind to either LF or EF. Okay, so binding sites for edema factor, which is EF, or lethal factor, which is LF. Okay, but it's not specified how many of each you're going to bring in, so it's not like you always bind 2 edema factors and then 1 lethal factor. It varies. Okay, right. So the other components of the anthrax toxin are now bound to this heptamer. Now, also what happens when you heptamerize the anthrax toxin protective antigen 63 with its receptor and co-receptor? What starts to happen is you're going to get depalmitylation of the tumour endothelial marker 8. Okay, so you're going to start removing these palmitoyl groups. Now let me highlight them up in orange. So remember, these were these palmitoyl groups which were attached to cysteine residues on the cytosolic domain of the tumour endothelial marker 8 protein. Now, what's going to start happening is these are going to be depalmitoylated. Okay, so they're going to be removed, basically. And whether this occurs spontaneously or whether enzymes are needed to catalyze this, I don't think is known. Okay, it's likely that enzymes probably catalyze it, but basically the binding of the PA63 and the heptamerization that this causes promotes then the depalmitylation of the tumour endothelial marker 8 protein. So once you've heptamerized, what's going to happen is the cytoplasmic portion of the tumour endothelial marker 8 proteins, which are all in this heptamer, and you're going to have 7 of them, they're going to start being removed, basically. So you're going to start depalmitoylating the cytoplasmic aspect of the tumour endothelial marker 8 once they've heptamerized like this. Okay, right. Now, what does this cause? Okay, so you've removed these palmitoyl groups now. What's the significance of that? Well, basically, this allows them to go into special portions of the membrane known as lipid rafts. Now, this is very, very odd, because these special portions of the membrane, which I'll explain in a moment, called lipid rafts, usually palmitylation of proteins promotes the protein going into the lipid rafts, but in this case the depalmitylation of the anthrax toxin receptor is what promotes the anthrax toxin receptor heptamer going into the lipid rafts. Okay, so what's going to happen is depalmitylation, and then you will go into the lipid rafts, and that is very odd. Usually you'd have to palmitylate proteins to make them go into the lipid rafts, so this is odd. Right, now let me take a step back after saying that and now explain to you what a lipid raft is. Okay, so basically, if we are looking at the outside of a cell, okay, and let's say we're looking down at a great big chunk of the cell membrane here. Okay, so this is all just cell membrane. Basically, the cell membrane is not just one homogeneous layer, basically. Instead, there are regions in it that are special. Okay, so there are little lumps of the cell membrane which are called lipid rafts, basically. Now let me colour in one of these lipid rafts in orange. And what's special about these lipid rafts is that they have different types of lipids within them from the normal cell membrane here. So this is the normal portions of the cell membrane, and the special fancy word for the normal portion of the cell membrane is to call it the glycerolipid region. Okay, so most of the cell membrane, the normal portions of the cell membrane, are made up of glycerolipids, and glycerolipids glycerolipid is the fancy word for any lipid molecule that is based on the structure of glycerol. Okay, so let me just remind you of the structure of glycerol. So the proper name for glycerol is propane 1,2,3-triol. And although propane 1,2,3-triol is a mouthful, it's useful because it tells you exactly what the structure of this is. It's a 3-carbon molecule where each of the carbons has an alcohol group coming off it. Okay, so this is the structure of glycerol. Okay, also called propane, since we are based on propane. Propane's just the name for a 3-carbon hydrocarbon, and then it's propane. Let me just pull this out a little bit. 1, because off the first carbon we have an alcohol group. 2, because off the second carbon we have an alcohol group. 3, because off the third carbon we have an alcohol group. And then triol, because we have 3 alcohol groups overall. Right, now, basically, you can take long-chain carboxylic acids, which are also called fatty acids, of which palmitic acid is an example, okay? But there are other examples as well, for instance, stearic acid, oleic acid, linoleic acid, all of these, okay? So, a fatty acid is just the old biochemist name for a long-chain carboxylic acid. It's a carboxylic acid that has a really long tail here, basically. And what you can do is you can esterify these long-chain carboxylic acids to the alcohol groups of the glycerol molecule to create monoglycerides, diglycerides, triglycerides, okay? So if you put a single carboxylic acid molecule onto one of these alcohol groups and leave the other 2 alcohol groups alone, that creates what's known as a monoglyceride. Okay, if you put 2 alcohol groups on, that will create what's known as a diglyceride. If you put 2 carboxylic acids esterified to 2 of the alcohol groups, that creates what's known as a diglyceride. And if you put 3 carboxylic acid groups on, 1 onto each of the alcohol groups, that will create a triglyceride. Okay, so glycerolipids are based on glycerol. And of course, the major component of the phospholipid bilayer is phospholipids, and phospholipids are basically, again, they're based on glycerol. Okay, so let me draw a little cartoon for a phospholipid. Okay, so this is the cartoon for a phospholipid. Basically, these 2 vertical lines, these represent the long-chain carboxylic acids. This horizontal line here, which is in green, represents the glycerol molecule, and you've esterified to the first and the second alcohol groups of the glycerol molecule long-chain carboxylic acids. And then to the third alcohol group, you have added a phosphate group, basically, and this is the structure of a normal, boring old phospholipid, and most of the cell membrane consists of these phospholipids. Okay, and this is an example of a glycerolipid. Okay, right. So, most of the cell membrane consists of glycerolipids, molecules which are based on the structure of a phospholipid. It will also have some monoglycerides, diglycerides, and triglycerides in there. Okay, lipid rafts are special because they are not, they don't contain lipids that are based on glycerol, okay, so they contain a high fraction of non-glycerolipid lipids, okay. So these are different types of lipid molecules, and they tend to associate all together, so they don't tend to mix in with the other glycerolipids. Instead, they all aggregate together to form these lipid raft structures. So they're like rafts within the sea of glycerolipids, basically. So, what sort of lipids are present within lipid rafts? Well, there are three major components of lipid rafts. Okay, so three famous sort of lipids that are in lipid rafts. Okay, so the first is what's known as glycosphingolipids, and I will explain what a glycosphingolipid is to you in a moment. Okay, and specifically the type of glycosphingolipid that is particularly highly present within lipid rafts are gangliosides. Okay, so a ganglioside is just a special type of a glycosphingolipid. Right, the other two major types of lipid that you have within lipid rafts are you have cholesterol molecules, a lot of cholesterol molecules. And again, I'll show you the structure of cholesterol molecules. And then a very similar molecule to cholesterol, you also have ergosterol in lipid rafts. Okay, right, so let's now see what these molecules actually are, because it's nice to know what we're actually dealing with here, what it actually means to have a lipid raft, and to know that you really need to know what these three types of molecule are. So, in order to explain to you what a glycosphingolipid is, I first need to explain to you what a ceramide is. And to explain to you what a ceramide is, I need to explain to you what sphingosine is. So we're going to start off with sphingosine. Okay, right. And sphingosine's proper name is 2-amino-4-octadecene 1,3-diol. Okay, so 2-amino and then 4-octadecene. And again, although this is a nightmare of a name, it's actually very useful because it tells you exactly what the structure of this molecule is. And then 1,3-diol, and it's actually not that complicated to read. It's a nice name, basically. It makes logical sense. So, we'll start off with this octadecene here. So this tells us that we're dealing with an 18-carbon hydrocarbon, and that it has a single double bond, and that double bond is off the 4th carbon, and therefore will go between the 4th and the 5th carbon. So, let's draw the first 5 carbons, because those are the important ones. Because off the second one we have an amino group, off the first and the third we have alcohol groups, between the 4th and the 5th we have a double bond. But then we don't need to show the other 13, do we? I don't want to have to draw out 13 other carbons, and basically it's just boring after this. You know, the other 13 carbons are just saturated with hydrogens. That's not very interesting. So instead I will use a shortcut, and I will draw a methylene group. I will put my brackets around it. I'll put a 12 there to denote that you've got 12 of them. Repeat this 12 times, and then I'll have to draw out the final methyl group as well. So this deals with the final 13 carbons, which are all just saturated with hydrogens. This is the 12 methylene groups, and then right on the end we've had to draw out the methyl group. Okay, so that's the boring bit dealt with. Now let's do the interesting bit. So, between the 4th and the 5th carbon, we have a double bond, basically. That's this portion here. So, 4-decene tells us that we're dealing with an alkene rather than an alkane. Okay, so we need a double bond here between the 4th and the 5th carbons. So, also off the 4th and the 5th carbons, you will then just have a single hydrogen, like so. Okay, so that's the octadecene bit dealt with. Now let's deal with 2-amino. So off the second carbon, which is this carbon here, you need an amino group. Okay, so there we have an amino group. Okay, I've just realised that I need to show something coming off that amino group later, and I've positioned it in an awkward position, but never mind. Okay, can't be helped now. So let me just highlight this up. So here is the 2-amino. So this portion here is the 2-amino. In fact, I'll box it. That'll make it look cool. Right. And then off the first and the third carbons, you then also have alcohol groups. So an alcohol group comes off the first carbon. That's this bit. And an alcohol group also comes off the third carbon. And then we just need to fill in everything else with hydrogens. So I'll put a hydrogen here, and then 2 hydrogens off here. And that now is the structure of sphingosine, or 2-amino-4-octadecene 1,3-diol. Okay, now, ceramides are based on the molecule sphingosine. Now, there is not just one ceramide, there are a huge number of different ceramides. So a ceramide is a whole family of molecules. Okay, and basically they're based on the structure of sphingosine, and what you're going to do is you're going to bring in a carboxylic acid molecule, okay, and it is not specified which carboxylic acid you bring in, and this is why ceramides are an entire family of molecules, because of course carboxylic acids are a whole family of molecules. So bring in me a carboxylic acid, okay, and now bind it to this amino group via an amide link, basically. So let me show you this now happening. So what you're going to do is take off this alcohol group from the carboxylic acid group, take off a hydrogen from the amino group, bind that to the alcohol group to create water, and then bind this carbon to the nitrogen. So let's now draw this structure out. So here is the second carbon. It has its amino group, which is now bound to this carboxylic acid group, so now is no longer an amino group, but is an amide group instead. And then everything else remains exactly the same, so let's just copy it out from above now. So the first carbon will have an alcohol group and then two hydrogens coming off it. The third carbon will then have an alcohol group and a hydrogen coming off it. The fourth carbon will then have this double bond to the fifth carbon, and both of them will have a single hydrogen coming off them. And then finally you will then have 12 methylene groups, culminating with a methyl group right on the end. Okay, so here are the 12 methylene groups, and then here is our methyl group right on the end. Okay, so this is the structure of a ceramide, and of course this R group is arbitrary, so there isn't just one ceramide. You can stick whatever R group you like in there, and you end up with a huge number of different ceramides. Now, these are all lipid molecules because they have this huge, great hydrophobic tail which will anchor them within membranes. Now, we haven't arrived at glycosphingolipids yet, but they're going to now be based on the structure of ceramides. So we're going up steps, basically. We went from sphingosine to ceramides, and now we're going to go up another step to glycosphingolipids. So basically, to turn a ceramide into a glycosphingolipid, what you have to do is take off this hydrogen from this alcohol group off the first carbon and replace it with an arbitrary R group here. So the hydrogen goes, and it's going to be replaced by this arbitrary R group. So get rid of that hydrogen there. Okay, and when you do that, and I should just stress that this R group doesn't need to be the same as this R group, so I'll put that as an R prime group. Okay, when you do that, you've now got a glycosphingolipid. So glycosphingolipids have two degrees of freedom, if you like. Here is one variable here. You can vary this R group, and here's another variable. I should use variable rather than degree of freedom. Here's another variable, R prime here, which can again be anything. Okay, so glycosphingolipid then now. So this is what is meant by a glycosphingolipid, and these are the sort of structures that you have sitting within lipid rafts. Now, gangliosides are a specific type of actually, let me just make a little correction here. I've made up something rather foolish. This structure that I've drawn here isn't necessarily a glycosphingolipid. This is what you would call a sphingolipid rather than a glycosphingolipid. So take the glyco off. This is actually called a sphingolipid. Okay. Now, glyco, if this R prime group here is carbohydrate in nature, then you would give it the prefix glycosphingolipid. But if it's just a generic R prime group here, then it might not be carbohydrate in nature, and then it would just be called a sphingolipid. I apologize for that. Okay, so I hope that hasn't caused confusion. This molecule that I've now drawn here, where you have two variables here, would be called a sphingolipid. If this second R group over here is a carbohydrate R group, i.e. it's based on monosaccharides, then you would call it a glycosphingolipid. and gangliosides are specific types of glycosphingolipids. So gangliosides will have this basic sphingolipid structure. Then in this second R group they will have a carbohydrate group, and this carbohydrate group will have a specific type of structure, so that this is then called a ganglioside. So gangliosides are specific types of glycosphingolipids. I apologise for any confusion that might have caused. Right, okay, so we'll call it there for this video, and we'll continue our discussion in the next video, where we'll turn our attention to what cholesterol and ergosterol are.